Docking is simply is going to find out the interaction between two components. So that is what the basic concept of docking. So we are going to take two components. One component should be a target, another component should be a ligand. Usually the target, in most of the studies, the one particular target is going to be usually a protein. The other one component will be like a chemical substances, small molecules, or rarely it might be another protein molecule. So docking usually like protein ligand docking or protein protein docking. So this is what the different types of docking generally carried out. And then one more thing is that in docking studies for protein ligand docking, different tools are available. Actually, in the course I discussed about PatchDock server, but at present time the PatchDock server is not working fine. So today I'm going to explain the protein docking using the SWISSDOCK server. Okay, so now we will go for SWISSDOCK server. So for before going to this docking server, we need two components. One is protein, another one is ligand. Okay, so for protein, we have to go for protein databases to collect a protein. We can use it for docking purpose. Similarly, for ligand, we can identify the ligands from the natural resources or the available databases from the PubChem. We can take any chemicals from the PubChem, or we can collect the chemicals from the GCMS analysis of any plant sources. Okay, so two things are required. So first, for today's class, I'm going to give an example for identifying the inhibitor for the enzyme. So it is going to be oxalate oxidase enzyme. So we'll go to this to docking studies. That is what here 2RDU. So this is what the ID for the human oxalate enzyme 2RDU. So this is the human glycolate oxidase enzyme. So this is bounded with the glyoxylate substances. So this is the protein we are going to use for our docking, today's docking purpose. And this enzyme is going to contain the chain A protein sequence, and then it will contain two ligand molecules. One is a FMN, another one is the glycolic acid, glycoxylate. So one is a glyoxylate, another one is a FMN. So these are the two ligands which are present in the protein. So that is what we are going to use in the docking studies. And then with respect to ligand, we are going to take chemical substances derived from the plant Arva lanata. So GCMS analysis of Arva lanata we have carried out, and in that GCMS analysis we got the presence of this hexadecanoic acid. So I am going to use this SMILE file from the hexadecanoic acids of Arva lanata GCMS study. Okay, so now we have a protein structure and we finalized the ligand structure. So now we go for a docking. We are going to use SWISSDOCK. So this is the present SWISSDOCK server. The old server is actually available here. This is a new server. So in this server, first they are asking a ligand structure. So that is mostly in the SMILE format. So we are submitting the SMILE of a chemical, that is the ligand. So once you paste this SMILE, you should click this prepare ligand option. So the server analyzes these available or provided SMILE structure. Once it accepts the SMILE structure, here you will see the tick mark. And then in second case, if you having a downloaded prepared PDB file for docking, then you can upload the file. Or if you know the ID of the PDB file, you can give the PDB file ID here. So this is what 2RDU is the file name, we know the structure of 2RDU. So we are submitting the ID here. Once you submit the ID, the SWISSDOCK server itself loads the PDB file, and then it will ask which is the chain I have to keep, and then which is the heteroatom I want to keep for docking purpose. So for the docking, it should not have any ligands. It should have only the protein part to be present in the form of target. So we are not allowing any ligand to present in the PDB file. Next, we have to click this prepare target option. So now you can see the target is also prepared. And then third option is that it will ask the box for center. Center means where the ligand is going to interact with this particular protein. So this is what defined by this particular box. So by default, since already this particular protein contains a ligand, the software identifies the box. So if the protein does not have any ligands, so or if we are interested in a particular place for docking, then we have to define this particular box, that is the box where the drug molecule or the chemical is going to dock. You can see now, so as per this box says, so the interaction point of the chemical structure is going to be in this particular box. Okay, so how this box to be identified from the protein structure? For that we need a software known as Discovery Studio. So now we can open, and I have already downloaded the 2RDU program. Okay, so I hope everybody knows it. We can download this PDB file by using this legacy PDB format. If you click, you can download this RDU file in a PDB format. So we have to open that downloaded file. By using this Discovery Studio. Okay, this is the protein. It is loaded. Then you have to go to this chemistry or view option hierarchy window. So here you can see this three component. One is the peptide chain part. The second one is the ligand part. You can see now the ligand is selected, and third is a water molecule part. Okay, so this is what preparation of the protein means. You have to remove the water and we have to remove the ligand. Okay, so during this part, in order to identify this particular box, okay, for the values, okay, you have to select the ligand. So now we have select the ligand. You can see this yellow color, the ligand is selected. Then there is a option known as, you can see here, the binding from ligand. Now you can see this, define and edit the binding site option. So we have to select this define editing binding site option. Then from current selection, you can see this from current selection. Once again I'm telling, you have to select the ligand, and then in the receptor ligand interaction option, there is option known as define and edit binding site. In that option, you have to select a ligand. After selecting the ligand, you have to select this from current selection option. If you select this, you can see the formation of one ball over this particular ligand. So this is what the location of the box is indicated. So now you get the value of this box from this attribute options. You can see now the attribute option is available. So it is around 27, -9 and -15. So you can copy this value and you can provide this value here. So around 27, and you can see this is around 9. You can see this is around -9 and this is around -15. And then we can also reduce this box like 15. Even if you allow 15 or 20 it's okay, so we can reduce it as 15. So now you can see the box size is reduced, and you can check the box still is present within the protein sequence. Then you go for this select parameter option. If you want more number of clusters, you can increase the clusters. It's better recommended to have a minimum number of clusters so that you can get a easy, quick result. So now you click this check parameter. And then once you get this particular check parameter option is also positive side like green tick, then we go for this docking. So for that we have to give email ID and title of this particular docking. Okay, it will take a little bit of time. So now you can give your mail ID to which you should get a result. So I'm giving a mail ID and then title. So I'm giving test session. Already I have tried it. So I'm adding test session 1. Then I'm clicking this start docking. So now it is going to start docking, and it will give a link also. So this link is going to upload the results, will be available here once the docking is complete. And we also we will get a mail from the SWISSDOCK server once docking is complete.